So for our second talk, we have Margarita, who's from the University of Birmingham. We met briefly at the BECMC, which I would recommend that everyone go to, and our talks were on at the same time. So I've invited her here because I missed it then. I'm really looking forward to this.
True. Okay, hi everybody. Thank you very much for the invitation. So my name is Margarita. I'm a third-year PhD student from the University of Birmingham. And today I'll talk to you about my model, which looks at how spheroid growth is affected by ECM stiffness. Hopefully this works. First, I just want to acknowledge my supervisor Fabian, co-supervisor Pradeep, and also my collaborators, Susan Cox's lab and John Metzger, that work with me. Okay, so I will start with an overview of the problem, and then I'll go through the actual model and the results that I've got so far. So what is spheroid growth? So when we talk about spheroid growth, we're looking at a group of cells which are grouped together in a sphere shape. Usually this is about cancer cells. So it's a little tumor in the shape of a sphere. And this tumor is surrounded by its tumor microenvironment, which is mainly composed by extracellular matrix. So the extracellular matrix, or ECM, is a complex network of numerous macromolecules which are all linked together. The main component of the ECM is collagen, and this ECM is very important because it's present in all tissues outside the cells, and depending on the organ that we're looking at, it will have different compositions and structure. Yeah, that is all wrong, whatever. So there are some factors that affect cell behavior, mainly the phenotype of cancer cells that we're looking at and the mechanical and biochemical features of the ECM. So again, changing the composition and structure, the ECM will have different properties. So my model is based on some in vitro experimental data, and the question that we're trying to answer is, how does the ECM stiffness affect collective cancer cell migration, because it is a group of cells migrating, and in particular spheroid growth. So the experiments have been taken by Tom Phillips, and they looked at a spheroid of epithelial cancer cells surrounded by collagen only. So this is already a simplification of the ECM. And then to this collagen, they've added some ribose. What the ribose does is adding links between the collagen fibers, and this changes the stiffness of the overall network. And so by increasing the concentration of ribose, the ECM stiffness will increase as well. Okay, moving to the model. So my model is a hybrid discrete-continuous model. And I am developing the model in PhysiCell, which is a modeling platform, is a software used usually for just modeling cells, migration, and other behaviors in general. And in fact, in PhysiCell, the cells are modeled with an agent-based model, and they are modeled as little spheres. And this agent-based model is off-lattice and center-based. What this means is that the cells can move freely in the space, and their position is given by the center of these spheres. Then PhysiCell has a continuous microenvironment. So this continuous microenvironment is then discretized, but in PhysiCell, it is actually used mostly for diffusible substances. You can think of oxygen, for example. And it is possible to model the ECM using this continuous microenvironment. However, what I'm doing is using an extension of PhysiCell developed by John Metzger. And in this extension, again, we have the continuous ECM discretized in a mesh, so a grid, and each voxel, which is a 3D pixel, is a ECM element. So each ECM element will have information about the average fiber orientation, the anisotropy, which is how parallel the fibers are, and the density. And then to this, we add ribose concentration. So even though in PhysiCell it is possible to model in 3D, what I'm doing at the moment is a model which is a constrained 3D model. So essentially the cells cannot move in the Z direction, but the behavior that they have is still mimicking what happens in 3D because they are surrounded by the ECM. So you can imagine it as looking at a slice of our spheroid. So I split my model in 3 parts: cell movement, cell proliferation, and ECM remodeling. So for cell movement, we have 2 components that affect the cell movement. So the interactions between the cell and the ECM, and then the cell-cell interactions. I will go through those in a minute. For cell proliferation, there are some built-in functions in PhysiCell to define the cell proliferation, but I also added a contact inhibition or proliferation function that essentially is saying that if there's no space available, the cells will not proliferate. And then finally, for ECM remodeling, the interactions between the cells and the ECM will change the ECM elements' orientation, anisotropy, and density over time. So the way these interactions work, for cell-cell interactions, we're looking at the neighbors. So we're just saying, if we look at a cell CI, I'm looking at the neighbors around the cell, so just the cells within a certain distance from CI. And instead, for the cell ECM interactions, I use 2 different ways of defining the nearest voxel. So the first way is by looking at the nearest voxel to the center of the cell, so the position of the cell xi, whilst the second way is looking at the nearest voxel to the point on the membrane, so the surface of the cell, in the direction of movement of the cell. So essentially, I'm just looking at the point which is in the front of the cell, and the cell will interact with the voxel in the front. And that makes sense because, for example, things like degrading the ECM fibers happens often in the front of the cells through some protrusions and some enzymes that degrade the fibers. Cell movement. Right, so we said cell ECM interactions. In PhysiCell, this component of the velocity is called motility, and obviously is composed of motility speed and motility direction. The direction of movement on the cell depends on the orientation of the fibers and the anisotropy, and essentially the cell will tend to follow the orientation of the fibers, in particular if they're more aligned, so the anisotropy is high. And then for the motility speed, instead, the motility speed only depends on the ECM density. Through that function, that is called haptokinesis, and essentially it's just saying that the cell motility speed will be max for an ideal value of the ECM density. Okay. For the mechanics, so the mechanics represent the cell-cell interactions, and this is actually predefined in PhysiCell. So PhysiCell defines 2 different forces and 2 different potentials, an adhesion and a repulsion potential. So the adhesion potential wants to mimic how the cells with these little protrusions, they adhere to each other and they pull each other, like themselves, I don't know how to say it, like they pull the neighbors towards them. And in fact, the radius of interaction is RA, which is larger than the radius of the cells. The interaction is only when the cells are in contact, and it's not a proper force, it's just trying to mimic the space occupied by the cells. So essentially, they can't be compressed too much by neighboring cells. Important here are the parameters, obviously, because I have to tune the model with the data. And so the parameters that I will talk about later will be the maximum motility speed, S max, and then the adhesion strength, C adhesion, and the repulsion strength, C repulsion. For cell proliferation, so the division is quite simple. There is a certain probability for the cells to divide, and when the cells divide, they split into 2 orders of volume, which is half the initial cell volume, and then over time they will grow in volume again and divide, etc. What I've added is this contact inhibition or proliferation function, which affects the rate of division of these proliferation. So essentially, what I'm saying here is that I'm fixing a threshold above which for the neighbors, above which the cells will not proliferate at all. So this threshold is just, if I have more than n max cells around me, n max or more, then there's not enough space, the cell will not divide. And it is a decreasing function for ni less than n max. Similarly for the ECM density, if the ECM density is particularly high, then the rate of division will decrease. Remodeling. So I'm not going to go through all of these equations, but essentially how they work is there's just an description. So for the orientation of the fibers, they will change depending on the direction of movement of the cells. Anisotropy increases proportionally to the cell speed. What is interesting is actually how the density changes. So the density of the ECM changes towards a target value. When the ECM density is above the target value, the ECM density, the fibers will be degraded, and so it will decrease. When the ECM density is below this target value, the cells will produce more ECM, and so it will increase the ECM density. What I've added here is how the rate of this deposition and degradation of the ECM depends on the ribose concentration. So here I'm just saying that if the matrix is soft and the ribose concentration is low, the cells will degrade the ECM density faster. Well, the change in the ECM density will degrade the fibers faster, and if the ribose concentration is high, so the fiber stiffness is high, then the rate will be slower. Oh, and that's the only function in my model that has a direct dependence on ribose, and that has an effect on the overall behavior, because if I'm slower degrading the ECM, I will move slower, I will proliferate slower, and so it will have an effect on the actual spheroid growth. So some results. First of all, how do we measure how much the spheroid is growing? So on the right-hand side, you can see an example of a simulation. And the way we measure the spheroid growth is just by looking how the area covered by the spheroid over time. And what I did was just fixing a grid with the side of each square being roughly I think it's just the diameter of each cell, and counting the squares, just as simple as that. And that's actually kind of similar to what happens with biological data as well. They compute the area covered by the cells. And what we see on the left-hand side is what happens to the spheroid, the area covered by the spheroid over time. So 5,000 minutes is the time of a simulation, and we can see that for ribose 0, we have that the area covered by the spheroid over time is higher than 50 and 200. And this is very good because it's actually what the experimentalists see as well. So that was very exciting. So from now on, though, we will not look at actually what happens over time. We'll just find a ratio between the final area, the area covered by the spheroid time 5,000 compared to the initial time. And so we will see if it doubled, tripled, etc. So I have a lot of parameters, and I have to do some parameter analysis. So the first cool thing that I found was by changing adhesion and repulsion. So on the x-axis, we have adhesion and repulsion coupled together, and we see how much the spheroid grows, as I was saying before. And here in particular, you can notice that the ratio between adhesion and repulsion is always the same, is of 0.4. And that's because it means that essentially the equilibrium distance between the cells is the same for all these couplings of adhesion and repulsion. And what's cool is that even by fixing the ratio, when we increase both adhesion and repulsion, the area covered by the spheroid will decrease. So it migrates less, it invades less. So the next step is looking at, for other ratios, what happens. But first, this is what it looks like. So on the right-hand side, same video as before. with adhesion for repulsion 10. On the left-hand side, adhesion 10, repulsion 25. And this is very cool because like if we're looking at two different types of cells, a non-invasive type of cell and a more invasive type of cell. And certain experiments kind of look like this. So for me, this was very exciting when I found it. Now, I'm not sure if this is the right way of defining non-invasive and invasive cancer cells, but still pretty good. And the other cool thing is like these little branches there naturally form in the spheroid. And finally, yeah, the color just indicates how many neighbors they have. So unfortunately, when I ran the simulation for other ratios, so this one was the ratio that we were looking at before. For lower ratios, it doesn't quite work. I was expecting a gradient for all the different ratios, and this is not what happened. So it might be that the range of the parameters is not correct. I don't know. I still have to actually interpret these results, but definitely doing a study like this can help me figure out what adhesion repulsion values make more sense for the type of model that I have. So maybe I don't even have to look at ratios which are lower than 0.4. Maybe what I was looking at initially was good enough. And then another thing that I looked at is max motility speed. I guess this is quite intuitive. It's just saying that if the velocity term, which is the motility, which is the cell ECM interaction velocity, if I increase the maximum motility speed, then the spheroid will grow faster. So that's not a big surprise, but you know, you never know. And what's good again is the fact that ribose 0 is above 50 and above 200, even though for smaller values we don't have. But that again could also be an extra parameter to change for non-invasive type of cancer cells. So still interesting. And for future work, I just want to keep doing this parameter analysis, so maybe explore more about adhesion repulsion, max motility speed, but then also other parameters. So for example, proliferation rate, degradation rate, and deposition rate, and realignment rate, and see what happens by changing those. But also what would be very cool is to look at simulations with non-random fiber orientation initially. In fact, there are some experiments that look at how the fibers being more aligned with each other have a huge impact on spheroid growth and in general like cell migration. In fact, if they're more aligned, they tend to migrate more. And I did a simulation, it kind of worked, but I don't have it properly prepared. So yeah, I want to explore that further to see if my model also mimics that. And then the other thing that I would like to do is changing the functions that I already have just slightly. For example, one thing that I want to change is the contact inhibition or proliferation function and making dependent on ribose as well. And that's because other experiments also show that the stiffness of the ECM enhance proliferation. So for stiffer ECM, we would have more proliferation. So I could get a biphasic behavior, which would be very interesting to look at, in particular because the Susan Cox's lab, they found that high stiffness lowers proliferation, lowers migration, but other experiments see the opposite. So it would be good to find something biphasic that I can explore and see when that happens. And this is all from me. Thank you very much.